It's time. Hello, everyone. This is Chatan Noir, and you are listening to Into the Liminal Abyss podcast. And on this week's show, we have Dr. Jeff Meldrum. One of the most fantastic speakers that I have ever listened to, and I'm sure you guys are going to find him interesting too. So we'll just get started with the interview.
Okay.
Could you tell me what your scientific background is?
Well, I have a doctorate in anatomical sciences with an emphasis in physical anthropology. So that simply means that rather than the traditional four subdiscipline approach to anthropology, there was a growing need for gross anatomists in medical school and dental school and so forth, allied health professions. And the supply of anatomists was growing thin with the molecular revolution, work with DNA and cell biology. And so departments cast their gaze elsewhere for anatomists and found them amongst physical anthropologists. So a new track sort of arose. So, my first 2 years of graduate school were essentially medical school without the clinical side of it, and my interests focused in vertebrate functional morphology generally, but specifically locomotion adaptations, how animals, and particularly primates and humans, are adapted to their means of getting around through the environment. Bipedalism, in the case of humans, then being probably foremost in that regard. And so the fossil history of our evolution of bipedal adaptations has been the focus of my attention.
Excellent. And then, so with your extensive educational background, what brought you, or what pulled you into the field of Bigfoot? What piqued your interest in?
Sure. Well, my exposure began when I was rather young. I mean, my first contact with the subject matter came when I was in 5th grade. And the kids at school were talking about this program that was coming to town, and I ran home and opened the paper, and sure enough, there was an advert for Roger Patterson coming to the Spokane Coliseum with his documentary film, which showcased the 60-second clip that he captured on Bluff Creek. And the notion, this was my first I'd ever heard of Bigfoot, but the notion of a giant hairy ape, a bipedal ape, lurking in the woods of the Pacific Northwest was just fascinating to me. And embodied all those interests as a young person that I had in, I went through the dinosaur phase, and that led into human evolution, fossil man. I was fascinated by the fossil record of human evolution. I was fascinated by primate behavior and anatomy and so forth, and comparative anatomy. And so that interest waxed and waned. I mean, there were times I was extremely immersed in that and became, devoured John Green's books and Ivan Sanderson. I learned geography and biogeography studying Ivan Sanderson's book from cover to cover. And but other interests, would displace that as you go along. It was always there in the back of my mind, and then as I pursued my academic career, and those interests became quite focused in mainstream questions of anthropology. I was in a position to evaluate evidence. I'm going to wait for just a second.
I don't even know where that's coming from. I don't think there's anything. lightning.
it's pipes. Okay.
Okay.
Sorry.
I was gonna say,
I'm sorry.
it's not thundering
You had
and
it, please,
lightning
I assume.
outside.
Well, yeah, I thought it was maybe a maid with her cart rolling down the hallway. So, so while I had these interests in Sasquatch were there lurking in the background, my interest in human evolution and human bipedalism became quite focused. So when I was in a position to actually consider some of the evidence for Bigfoot, which was the footprints, those iconic footprint casts, captured my imagination, and then in 1996 I had a chance to actually examine footprints firsthand in the ground, fresh, extremely fresh, in southeastern Washington, just outside of Walla Walla. And knowing what I knew at that time, I knelt down and I could see skin ridge detail. I could see the signatures of a dynamic step, tension cracks and pressure ridges and drag-ins and drag-outs and flexed toes and extended toes and slippage. And, these were not just static rubber stamped prosthetic fake feet that had made these. And so the hook was set, more or less. I mean, there was no getting around it. I can remember kneeling there beside one of those tracks, thinking to myself, being familiar with what Grover Krantz had gone through in his academic career for his interest in this subject matter, asking myself, do you really want to go down this road? And because it was a fork in the road. Either I just thought, oh, this is interesting, and went on my merry way pursuing my academic interests as they were, or I would begin to devote attention to this question and try to extend the work that Dr. Krantz had initiated and apply my more extensive knowledge, more specialized knowledge of foot form and function to this question of footprint evidence, in spite of the potential consequences. And here I am. So obviously the result of that little debate, that mental debate, is all too obvious, and the consequences have been all too obvious too.
Yeah, I know you've taken some punches on that one, but you're still here, and we love you for it.
Yep.
Now, how often do you actually get out in the field and actually go looking on your own for evidence, or is it just you get a phone call from someone and they say, hey, I think I have possible tracks?
Well, it's both, but definitely the former as well. I used to do much more. There had been a little bit of a hiatus. Our principal goal in going into the field, in addition to looking for trace evidence and physical evidence, was to try to collect DNA. I mean, I saw the demonstration of a novel species on the basis of a new DNA sequence as the way to go, as opposed to advocating the lethal collection of a type specimen. When a couple of investigators, first Melba Ketchum, Dr. Melba Ketchum, veterinarian from Texas, claimed to have DNA sequence from numerous samples that she had received from amateur investigators. Well, I thought, well, let's let this play out, and while the analysis and study is being conducted, we'll redirect our energies. So rather than spending a lot of time in the field, I undertook some other projects, like establishing the Relic Hominoid Inquiry, an online scholarly journal, developing a digital archive for the footprint evidence, using 3D scans that could be accessible by anyone that was interested, in open access way to disseminate that data, make it available to other researchers. Unfortunately, Dr. Ketchum's undertaking was a sham, and I was excited and relieved when Dr. Brian Sykes, an Oxford molecular biologist, undertook a project. His attempts to cooperate with Dr. Ketchum were rebuffed, which in hindsight now doesn't surprise me in the least. But even Dr. Sykes didn't have the best approach, and was driven by other goals in the media, his book writing and his documentary production. And I offered to screen the samples that were coming his way so that the effort would be focused
Right.
on the most likely to produce positive results. Because you can stick a hair under a microscope and examine it, and on the basis of the morphology determine whether it's a primate hair or not, or whether it belongs to some other form of common wildlife. And my experience had always been, when people would submit hair samples to me, that the majority of them were misidentified. I mean, they were just common wildlife. No, he said he didn't want to impose some presupposition about the nature of Bigfoot hair. And so he threw the barn doors wide open and accepted all these samples, and
That's a lot of wasted time.
it was a
and
lot.
energy.
And not surprisingly, the results spanned the gambit from human to porcupine in
Right.
Texas. I could hold a porcupine guard hair up to the light right
Yeah.
here and tell you it was not a primate hair.
Right.
So it was very disappointing, and there seemed to be many samples that went unaccounted for.
Oh.
My fear was that we had a repeat of what had been the experience in the past, and that was one of the characteristics that distinguish Sasquatch hair is the lack of a cellular medulla. So a shed hair that doesn't have an active follicle or a skin tag attached to it has very little DNA. There's very little
Right.
nuclear or cellular material. The cortex of the shaft of the hair is extracellular protein that's exuded, and there is no mitochondria or nuclear fragments to yield DNA. So my fear is that the samples that did not yield DNA went unreported in the final paper. I can't confirm that, and haven't confirmed it with wouldn't expect to confirm it with Dr. Sykes. But anyway, so Dr. Sykes. So long answer to your question. Those two projects sort of diverted my energies away from the DNA and field research for several years. Now I've tried to pick that back up. I'm working with some investigators, with one group in particular, Big Search out of Omaha, Nebraska, and we're undertaking a number of different initiatives, including an aerial survey approach, which involves a 25-foot helium-filled drone, with which we hope to equip with thermal imaging cameras and do aerial surveys at night of our study areas, and cover a much larger area than we could on foot or in vehicles.
Yeah, that's the thing with the thermal imaging is just to on a plane level try to look through the trees to find a heat signature.
Right.
You're only going to see maybe the first 2 trees in, nothing
Right.
past that.
Right.
But from the aerial, you've got a better chance looking down of seeing a heat signature
Right.
off of something.
There's still the problem of penetrance of the canopy,
Yes.
but the tree, in coniferous forests, the tree crowns usually aren't interwoven.
Right.
And so there are gaps. And, we'll have to be very selective in our sites so that the forest is thinner, not as dense, and patchier, and lots of interface areas at lake margins and streams and open south-facing slopes of ridge lines and so forth where we might have a better chance of counting. So it's not, it won't be a 100%
Right.
exhaustive survey.
Oh, yeah.
But still, we can cover a heck of a lot more ground
Yeah.
and work in tandem with ground exploration crews to site verify any contacts or any suspicious heat signatures we find.
And that's pretty cool.
Yeah.
Right there. So now I consider you a expert on Bigfoot footprints and casts. What are the biggest differences between a human footprint and a Bigfoot?
Right. Well, the most obvious one, obviously, is the size.
Yes.
So, adult male and female Sasquatch run from anywhere from like 14 inches to the largest credible track I have in my collection is 19 inches.
Okay.
And there's a bit of bimodality there between males and females, with some overlap, as you might expect. I mean, to the degree we can ascertain whether they're male or female, there seems to be a pattern that the smaller ones are also more gracile, and the track, and that fits the pattern we have from the tracks left at the Patterson-Gimlin film site. Patty's foot was rather narrow compared to what I think was the male in that territory at that time, which was the Jerry Crew and the Bluff Creek 1964, the Patterson, and it shows up other places as well. Chaim Baum and Bob Titmus cast that same individual, but he has a much wider foot relative to foot length. So the size is different. The other most striking distinguishing characteristic is the presence of a longitudinal arch in the human foot, and what goes with that, the raised arch, and then the concentrated pressure beneath the heel, then the heel bone, the calcaneus, and primarily the ball of the foot at the base of the big toe. So the presence of a well-differentiated ball and that hourglass shape and a well-differentiated heel pad distinguishes the human from the Sasquatch, which is relatively flat, very flat. I mean, there's some topography, but it's essentially flat, archless, and retains a greater degree of flexibility through the midfoot, which allows for the foot to conform to irregular surfaces. It allows for walking up steep inclines much more readily. It spares the toes, the bending stresses that human toes experience when weight is concentrated through the ball and during the final toe off of our distinctive sort of gait, striding gait. In Sasquatch, the final impulse is passed through the entire forefoot, and hence the toes can still remain relatively long, not as long as a chimp's, but sort of intermediate between that of a early Australopithecine and a human, for example. And that gives greater prehension when you have to navigate up irregular surfaces.
Yeah.
So, I mean,
when you're going from rocks to sand to the logs to,
What's fascinating is the foot of the Sasquatch is not just simply a facsimile of an enlarged human foot. It shows distinctions that are biomechanically sound and elegantly appropriate to the habitat, the terrain that they're navigating through for a large, heavy bipedal primate. So it's remarkable. Most people, especially back in the 60s and 50s, no one had that understanding of the biomechanics of the human foot. Well, a few people maybe did. There were some orthopedic surgeons and so forth. There are some classic books on human foot function that go back to the early turn of this last century, but Roger Patterson didn't know that stuff.
Right.
Bob Gimlin didn't know that stuff.
Now, what is your, like, when you're trying to distinguish, is this a real cast or a fake cast? What are like the things that pop out at you that you first look for when you're?
Well, you might almost say it's kind of like good art. When you see it, you know it. And by that I mean there is a degree of familiarity with what looks natural. So when I see a footprint that clearly isn't, the first step is to distinguish it from human. Of late, there have been many, many more reports of footprints that are in the 7 to 9 inch range, and they're almost invariably human. And one of the simplest giveaways is the result of our habit of wearing shoes. Most people have a little toe that's turned on its side and pushed inward. Sometimes you can even, the shoes have been so confining. It's like when you buy grapes, open a crate and grapes first come out, those spherical grapes have been pressed to the point that they're now little cubes and have ridges. Well, your little toe can get pressed up there to the point that you form a ridge and a callus where it's been pressed into a little peak or a ridge. And that's a dead giveaway, the presence of the arch, obviously, and the well-differentiated ball, the proportions of the big toe to the little toes with their much longer stems, because they have 3 bony elements instead of just 2 larger ones, as we find in the big toe, has a distinctive look. In contrast, the Sasquatch has these other characteristics we discussed with flat foot and toes that tend to be more subequal in size. But there's other aspects, the proportions, the articulation of the toes, evidence of the toe stems, the interplay of the toes with the soil and substrate. I mean, because we're obviously dealing with two challenges. One is misidentification.
Right.
The other is fakery, hoaxing. And so misidentifications are pretty straightforward. I mean, the only other thing out there with a relatively flat foot, at least a heel down foot, and 5 toes are humans or bears.
Mm-hmm.
And it's not really rocket science, and that was one of the reasons I wrote the field guide that I wrote.
Which is excellent.
Yeah, thank you. Which includes some of the basic distinctions that we're discussing here, and gives a really graphic visual representation. Anyone with a little practice and a little bit of time spent just simply looking, and you can do this too. Just go on the internet and call up as many pictures of bear tracks as you can find. Call up human footprints, and look at the way they look in sand and in mud and on hard surfaces, at the swimming pool, for example, and so on. And get that image in your mind, that search image, and then when you see a Sasquatch track, the distinctions should be very clear. So those are the ways. I mean, I have examples that betray features, artifacts of fakery. For example, a genuine track is left by an animal that probably weighs 600, 800 pounds. And when imprinted into the ground deeply, that soil is compressed and compacted. Usually, well, depending on the impulse, but a walking step will compact rather than impact and extrude, as when you're running with greater force of impact, acceleration of impact. But that's hard to do when you only weigh 100 to 200 pounds. So what fakers will do is use their hands and push in and exaggerate the margins.
Okay.
But that causes a displacement, and usually the displacement is towards the center. So you get a high center with a deep furrow outlining the footprint. And then how do you make toes? Well, your fingers are the closest thing to toes. So usually there are traces of some giveaway that fingers were used to make the toes, and then the articulation of the toes to the foot is never natural looking.
Right.
The way in which our toes articulate, are jointed to the bones within the instep, gives us a particular pattern, and the pattern of splay of the toes, involves movement at those joints, which are biaxial in a certain way. So they're going to converge to those articulations. They're not going to converge to some point back here in the heel, for example. So those are dead giveaways. So.
Okay. And now, has much research been done on? Because I know, as human beings, we have individual fingerprints, and I'm assuming toe prints.
Right.
Now, has any research been done to try to figure out, to identify key individuals, that tracks are associated with?
Right.
Is anybody doing their fingerprints and toe prints?
Right. Well, it's very limited because there are very few tracks that actually preserve dermatoglyphics. It's not a singular event, but it's a rare event. You have to have, first of all, you have to have a healthy skin surface, one that isn't too disrupted by extensive callousing or scarring or abrasion, which are things that can happen to a foot that is constantly exposed to the environment. 2, you have to have the transfer of those ridge details to the substrate, and that requires a substrate that has a fine enough particulate matter in order to pick up that fine detail. In other words, it's just like you have to have a big enough megapixels in your camera to actually see the details. Tiny little particles that will pick up that level of resolution. And that's not very common. It requires very silty soil. It requires soil that's dominated by glacial loess, which is that fine glacial pulverized rock that makes it like talcum powder. Or some logging roads, for example, that are dirt roads back before they really invested in creating quality gravel beds. the dirt would just get pulverized by these trucks to the point that every time a truck goes by, a cloud of
Yep.
very fine dust. That will pick up remarkable detail. But then it has to survive until someone discovers it. That very fine dust, not consolidated, every time a truck goes by again, dust rains down,
Yep.
or it literally jiggles it, like that scene on Independence Day when the spaceship
Yeah.
flies over the moon and the footprints get literally jiggled out of the dust, that would happen. Or the soil, if the soil is wet and conditions are very wet, like we have overcast and rainy skies today, the gravity and moisture, the content, will literally pull those ridges out and flatten them. I've actually done some experiments out in the garden where, I would I'd step and then wash my foot of any dirt and step again. In Idaho, we have a lot of loess, so the dirt in the garden is almost like a clay when it's not prepared with added organics. I made a series of in wet loess, very clear footprints which exhibited dermatoglyphics very, very clearly. And I poured a cast in one immediately, and then I waited 4 hours and I poured another, and then waited a few more hours and poured another, and marked them as such, and then carefully removed them and cleaned them once they'd set up. And you could literally then see over the passage of time the disappearance of those ridges. And after about 6 to 8 hours, depending on the weather conditions, depending on the moisture, etc., 6 to 8 hours, they'll be obliterated. And they only linger in places where we usually find, this is what's interesting, you usually find the very little traces of dermatoglyphics in a very spotty way in some footprints, and that's up along the vertical margins of the toes and walls of the track, which is interestingly enough. So the preservation, and then final step is you have to have someone who then makes the discovery while there still is ridges, and who is good enough at executing a footprint cast that they don't obliterate the dermatoglyphics in the process. Your plaster has to be just the right consistency to actually pick up and transfer that detail to the cast. So given all those variables, of my 300 footprint casts, there's only like about 6 that show extensive patches of dermatoglyphic. And of those, we have ruled out at least 2 or 3 as being simply pouring artifact, meaning the conditions were such, in this case very hot weather and a very pulverized road dust that has a wicking effect. It sucks the moisture. And when you do that, when you draw moisture out of the mixed plaster rapidly, it will set up, it will harden. And so you may have seen this when you're making pancakes. If you pour the batter on a hot griddle, it first hits and it sets, but then as the batter continues to flow slowly over, you get these alternating ridges, concentric ridges around the margins. Well, there were footprints where those concentric ridges were mistaken for coarse dermatoglyphics.
Okay.
And experiments had demonstrated that. And we did some such experiments with one of my students, using loess, varying temperature, and so forth to see what would happen under varying conditions, and these kinds of artifacts cropped up. So as far as identifying individuals on the basis of dermatoglyphics,
it's very hard.
It hasn't happened. So much more accessible and every bit as reliable, in my opinion, is the anatomy. There are lots of tracking books out there that talk about how, I was reading one just the other day, the Sand Bushmen were remarking how they can tell individuals on the basis of the shapes of their toes and other proportions. I mean, the appearance of the foot, but the real tell is the shape of the toes. And on that basis, I've recognized, as I mentioned earlier, this big male from Northern California back in the 50s, 60s, 70s. We have repeat appearances of this recognizable individual on the basis of the length of his foot, the proportions, the greatest widths, and the shape of his toes. They're very distinctive.
Cool. Because I was wondering if people in the field, I know with like the whales, they researchers take pictures of the flukes because each fluke
Yes.
is different.
Right.
And I was wondering if something like that applied with the North American Bigfoots, if, the individual characteristics of their footprint,
Yes.
if that's how you can tell, oh, this is the big male that I know from this territory, but what's he doing, 15 miles west, or,
Right. Absolutely.
So
that was one of the first questions I asked when I got involved with this, is I said, well, okay, now, because I had the impression that from thumbing through books, as just a sort of an avocational observer of this from the sidelines, thumbing through books and seeing a newspaper article, it seemed that every footprint was quite distinct.
Mm-hmm.
And I thought, oh, that's weird. If these things are as rare as they would have to be, then if you have repeat appearances of tracks in a given geographical region, the chances of those tracks belonging to one or one of few individuals should be really high. So we should see repeat appearances of individuals more often than a novel individual, especially in one geographical area. So I started looking for examples of that, and I mentioned the one of this individual in the Bluff Creek area back in the 50s and 60s. Well, I kept looking, and I found dozens and dozens of examples where we can see the same individual over and over and over again. There's some great examples from the Blues outside of Walla Walla because it's ideal tracking conditions. It's an area where many of the logging roads, the Forest Service roads,
Mm-hmm.
are unimproved. They're simply dirt roads. And so they make fantastic tracking beds. Any animal
Yeah.
that crosses the road leaves tremendous tracks. And so some of the investigators there have discovered what seems to be an inordinate number of footprints, but it's because of those exceptional conditions. One of the reasons I keep coming back there to do research.
Yeah. It's a hotbed
I
for
need
you.
that. I need that reinforcement.
Yeah.
It's like I'm a terrible fisherman because if I don't catch a fish in the first 5 or 10 minutes, I lose interest. But if I catch one right off the bat, I'm good for the rest of the day. I'll keep fishing and fishing and fishing.
So we won't see you on River Monsters anytime soon.
Probably not.
You'll be breaking the pole and throwing
Yeah.
it.
Well, I'll just be moving.
Now, I know that within the realm of Bigfoot, everybody thinks it's the same creature, but there's actually different species from all over the world,
Yes.
and different sizes. So, what do you notice are the differences in the footprints,
Yeah.
and is it because they're adapted to their climate that they're living in?
Yeah. Well, and their history and their
Okay.
phylogeny. So maybe to give that question context, is just 2 quick points. We have realized that the hominin, our human ancestors and direct relatives since the divergence from a common ancestor shared with chimps, as well as the hominoids, the other ancestors and relatives of the great apes, that the tree is very, very bushy, and it gets bushier and bushier with every new discovery.
Yes.
So we're finding a tremendous amount of taxonomic diversity. Plus, these discoveries keep affirming that some of the lineages on this very bushy tree, these terminal branches up here, have persisted much more recently, until much more recently than ever recognized before. So your listeners may think, oh, well, gee, one monster is one thing, but to have 4 or more, well, again, if you think of it in terms of the phylogeny, it's perfectly natural. We would expect. Why would there be just one when there's all these branches that have persisted so long? Why couldn't several different related forms be out there? So we've got the Sasquatch, we've got the Himalayan Yeti, which has, as you point out, distinctions in the foot, suggesting a divergent big toe. And it's a very stout foot, and obviously it's capable of walking on 2 legs. But a gorilla or chimp are capable of facultatively walking on 2 legs, for short periods of time. That retention of the divergent big toe, however, suggests that they still spend some time climbing trees. So the snowman isn't really a snowman. It's a jungle man that occasionally leaves tracks in the snow when it's crossing a pass from one valley to the next. But these valleys have subtropical forests in them, and so resources are in the canopy and so forth, and these animals clearly climb still. You have the orang pendek, which is probably a relic early hominin, like a late Australopithecine or very early Homo habilis, say. And so its foot is quite interesting. It has a non-divergent big toe. Let me back up. Homo floresiensis, which I think
the hobbit,
yeah,
for people
the
who.
hobbit. The foot is quite interesting. It has a non-divergent big toe, but it's very short. Its lateral digits are remarkably long, and its foot for leg length, the proportion is very high. So when it walked, it probably had, if you can imagine how you walk down to the ocean across the beach
Yes.
with flippers on. Many people turn around and walk backwards because you otherwise have to lift your feet up so high. Well, this creature had to probably lift its feet relatively high to clear those long feet, long toes relative to us. But a non-divergent big toe, just an early hominid. And then what else do we have? We have the Russian almas, which may be a relic Neanderthaloid, either Neanderthal or maybe a Denisovan. And its footprints look very much like ours. They are arched, but they're very broad, very stout, which agrees with the robusticity of the skeleton and musculature of the Neanderthal. But it apparently had, as suggested by the fossil record, our common ancestor with that would be Homo heidelbergensis, and that species was already showing evidence of an arched foot, a low arch in a very robust foot, but stability in the foot, shortening of the heel segment somewhat from earlier ancestors, which is an adaptation for speed, for running and distance walking.
Cool. Now, you just briefly covered if you were to give a guesstimate, how many different species do you think?
I'm comfortable with 4 right now.
4. Okay.
Which is remarkable because in 1961, Ivan Sanderson published a book and it also proposed 4. So that was 56 years ago. And of course, he ran into a brick wall because the paradigm in anthropology didn't allow for other non-human species to be contemporaneous with us. There could be only one species occupying the hominid niche through time. Mm-hmm. And so his thesis fell on deaf ears. But you read his descriptions, and I've written a second field guide which casts the net more broadly and considers this question of relic hominoids around the world. And that's the point where I arrived at too. And the characterizations have been modified just a little bit with the advent of, we have learned more and found more footprint evidence in the past 50 years. But essentially, my field guide reiterates the things that he asserted 56 years ago. It's kind of interesting how it's come around to that. I mean, there was a time when I really kind of shunned the notion because of the perception that that I alluded to your listeners maybe laboring under, that it's one thing to suggest one monster, one unknown, but then when you start multiplying that, it just gets a little implausible, ridiculous sounding. But now I've come around in my thinking and in the argument to a point where it actually is more persuasive that there is evidence of more than one, because of how we're learning, what we're learning about how the pattern of human evolution has proceeded, and that we should expect multiple terminal twigs on this bush to be persisting as relic populations alongside of us.
Because a lot of people, when you say Bigfoot or Sasquatch, it's an umbrella term.
Right.
Like when somebody says, "Oh, I have a dog." Well, yes, you have a dog, but what breed is it?
Exactly.
I'm researching Bigfoot. Okay, what species? What
Right.
what family tree are you chasing?
Right. Well, and there are some that advocate that even here in North America there are multiple species of unknown hominid, which I don't accommodate. I don't see the evidence for that. And the suggestion that there are different sort of, more ape-like footprints, the footprints are not credible and have been misinterpreted, in my opinion. The existence of three-toed and four-toed, in the excluding realistic anomalies or injuries or amputations, there's no basis for skinny foot with 3 toes or reptilian footprints, like the Honey Island swamp.
I was just about to say
which
the
is an
Honey Island.
alligator.
Yeah.
It's an alligator. Get over it. It's an alligator. The footprint, that is.
Yes.
And the hoax is undertaken by the individual involved in that are well documented, well known, and even the fake feet attached to a block of wood with a handle in order to press it into the mud have been observed and examined and so forth. So anyway.
So now,
Lyle Blackburn will explain all that to you.
Okay. Yeah, I'll be talking to him at some point. Okay, so one last question. I caught on two terms that you were talking about last night: the relic hominoid, and then you were talking about how you had basically pitched the idea of giving it a Latin.
Right. A Latin name or
Yes.
a nomen, yeah.
Yes.
All right, so two different things. There's a system called ichnotaxonomy that assigns scientific names
Yes.
to footprints, not to the creature. So that name, Anthropoidopes, I have to stop and get it right straight myself. Anthropoidopes ameriborealis will never be applied to Sasquatch.
Okay.
It's the name of Sasquatch footprints, period.
Okay.
And the diagnosis of that taxonomic name encompasses descriptions of the footprints. And that's a very useful exercise. It draws attention to this data set. It standardizes it, so we can take the Honey Island swamp monster and say, well, this isn't a Bigfoot because it doesn't have X, Y, and Z characteristics, which are the diagnosis of Anthropoidopes ameriborealis. Said it over here. So if it's genuine, it's something else altogether. All right. On the other hand, I've introduced this term, or I haven't introduced it. Dr. Boris Porzhnev, actually in Russia, introduced it, and I've resurrected it, and reapplied it, and tried to reinvigorate it, and that's relic hominoid. So a relic is a term which is used in biology, and I spell it with a T because that's the spelling that's used in biology, for populations that were once much more extensive but now exist in a much smaller range and in smaller numbers. It's a remnant is a synonym as opposed to an antique or a whatever, or something of religious significance, another application of that term. Hominoid has two potential connotations. In the more colloquial sense, hominoid simply means man-like. And we've heard the terms man-like monsters,
Right.
Monsters on Trial, the name of a book, and so forth. And so that's perfectly applicable. I intended, and I believe Porzhnev did as well in his use, in the more taxonomic sense, which means a member of the superfamily Hominoidea, which includes us and all of the apes.
Okay.
Chimps, gorillas, orangs, and even the lesser apes, the gibbons and sea monks. And the reason I prefer that over, say, relic hominids or hominins is that some of our relic populations are probably more closely related to the apes than they are to humans, like the Yeti of the Himalayas is almost certainly a relic ape. There's questions about whether Bigfoot is more closely related to something like Gigantopithecus, which, based on our current understanding of that species, would be more ape, or something like a Paranthropus. And we talked about, that one cannot escape the remarkable convergence or similarity between that skull of Paranthropus and Paddy on the film. In which case, then, it would be a hominin, relative since the divergence from the last common ancestor shared with us in chimpanzees. So either one of those, it works, whichever you're more comfortable with. If you don't know a lot about primate phylogeny, then it's just a remnant population of man-like species.
Okay. Excellent. I heard you mention it, and I thought, okay, I'm going to have him talk more about that in the interview, because that's fascinating.
Well, and the reason I'm advocating that, and as I said, Bigfoot clearly has a tabloidish stigma to it.
Yes.
It's just, Sasquatch I prefer because out of deference to the Native American reference, and it's a more descriptive term in the sense of it means the wild man of the woods.
Yes.
and there certainly is a precedent for that. Orangutan means wild person of the woods, the wild being of the woods, wild man of the woods. And Sasquatch has though been so overused that even now it's almost, absolutely on par with Bigfoot. And so to avoid that stigma and to provide a term that can be used by people who want to discuss it strictly in scientific circles, in scientific terms, the relic hominoid is a term that people can deal with. Academics,
yes.
Like I said, some of my academic colleagues are much more willing to discuss the prospect of surviving species of hominoids, in terms of relic hominoids, as opposed to one in particular. and so that's the goal. I mean, that's the goal. So I encourage people to use that term. There was recent recognition. The New Scientist magazine, which is the British equivalent to, say, our Discover magazine, semi-popular science magazine, had a cover story called "The Top 10 Puzzles, Top 10 Mysteries of Human Evolution Today," facing researchers today. And number 9 was, "Are other hominids alive today?" Now that was a huge concession.
Oh yeah.
That, 20 years ago, you never would have seen that in print. So that was an acknowledgement that, hey, this is a question that needs to be addressed. And it didn't get the associate editor who wrote the article didn't do the best job, I must say, unfortunately. So it didn't really get the type of exposure I would have hoped for. But nevertheless, it's there, and I can refer to it, and I can draw attention to it, and others have written about it as well. So other editorials and whatnot. So there's progress being made.
Excellent. Well, I know our time's getting short because we both have other things to get to today, but